So genetic testing for inherited breast cancer started way back in the early 1990s when the location of inherited predisposition to breast cancer was mapped to chromosome 17q21, and a hypothetical gene, BRCA1, was named by Mary Claire King. 4 years after that linkage analysis paper, the BRCA1 gene was actually identified. And then 2 years later, BRCA2, its sister gene, was subsequently identified. In the 2000s, we had discovery of additional cancerous genes, such as CHEK2, ATM, and PALB2. And these were identified either through linkage analysis or candidate approaches, or in some cases, large case control analyses. In the 2010s, we had widespread implementation of short-read-based gene panel sequencing for these known cancerous genes and many other cancerous genes. And looking at that data, both patient data and variant data, we now have a much better understanding of cancer risk by gene and by variant type. So we now think of there being 10 genes for inherited breast and ovarian cancer. And this is a table adapted from a review by Will Folks in 2021. We do, of course, have the high-risk, well-characterized genes like BRCA1, BRCA2, and PALB2. And then there are some other syndromic genes, such as P53, which can cause Li-Fraumeni syndrome, which is a very rare tumor syndrome characterized by early-onset breast cancer and predisposition to sarcomas, among other cancers. There are also two genes called ATM and CHEK2, which have moderate or intermediate effects, and these are predisposed about a 2- to 3-fold lifetime risk for breast cancer and some other cancers. And then finally, we have a group of 4 genes, BARD1, BRIP1, RAD51C, and RAD51D, which predispose to high risk for ovarian cancer and triple-negative breast cancer, but seem to confer a moderate risk to estrogen-receptive breast cancer. So clinical recommendations for patients with germline damaging mutations or pathogenic variants in these 10 genes have now been widely established and in use in the clinical setting. And these include enhanced screening and surveillance techniques such as MRI and surgical and treatment options. For BRCA1 and BRCA2 pathogenic variant carriers, they are eligible for PARP inhibitor trials if they've been diagnosed with breast, ovarian, prostate, and even pancreatic cancer. And some of the other genes on the table here are also involved in the same DNA pathway as BRCA1 and BRCA2, so they may actually become relevant candidates for PARP inhibition trials too. So Marie Claire has been collecting severely affected breast cancer families since the 1980s, and here are 5 examples of some of those most severely affected families. Each filled symbol represents either breast or ovarian cancer diagnosis with the age of onset listed too. And as you can see, in some of these families, the breast cancer has been in 2 or 3 or even 4 generations of the family. Over the years, we've applied short-read sequencing technology to these and other families, including exome and panel-based short-read sequencing. But we do have many families that have not been resolved, in that we haven't seen any pathogenic variants in any of the known breast or ovarian cancer genes. So some of the potential mechanisms that may explain these families that are not routinely currently tested include regulatory variants that could be upstream within open chromatin, either at enhancers or promoter regions, or open chromatin. And a variant such as a SNV or a structural variant within a potential enhancer may prevent transcription factor binding and either reduce or silence gene expression downstream. Another mechanism that's not routinely detected by short-read approaches are the mobile element insertions, such as ALUs, line elements, or SVAs. And even when they're inserted in introns, they can end up getting spliced into the normal message of the cancer gene, leading to reduced or inactive gene expression. And then finally, there are also deep intronic variants that are not routinely detected by short-read panel-based approaches. And here, these can either be point mutations or small insertion deletions that can either activate, strengthen, or create new cryptic donor or acceptor sites, which then allows for a pseudogene to be expressed, and often it's integrated into the normal BRCA1 or BRCA2, for example, message, leading to mutation through either truncation or gene splicing effects. So I'm going to concentrate today on the deeply intronic variants. Oh, and I should say, of course, that all three of these mechanisms are readily detected by long-read sequencing in a single experiment, and of course, that's adaptive sampling. So this is the adaptive sampling pipeline in our lab for breast and ovarian cancer gene testing. We do sonicate our DNA to about 15 kb and add barcodes during the ligation step. We target the 10 genomic loci, 1 megabase per gene region, so 10 megabases in all. And we're currently loading 4 barcoded samples on our R10 flow cell. This is output from a recent flow cell with 4 breast cancer patients. You can see we're getting median on-target coverage of about 25 to 40x. The on-target read depth, read length, is about 10 kb, and we're seeing close to 4x enrichment over whole genome sequencing. Our off-target reads are coming off around about 800 to 900 base pairs at present. So for this project so far, we've sequenced with adaptive sampling 240 relatives with breast or ovarian cancer from 120 families that haven't been resolved by short-read sequencing approaches. And as I mentioned, we have 25x coverage for each of the 10 genomic regions. Once we've identified all the variants from the long reads, we then do a filtering step. And here we're using the cancer-free participants in NOMAD-3. There are 75,000 participants in that study that do not have cancer. And we thought that was a good filtering group. And we do filter for rare variants, so we exclude anything that's any variants in BRCA1, BRCA2, or the other genes that's present on NOMAD V3 more than 3 times. And we were a bit more relaxed for the moderate penetrance genes and kept anything that had less than 10 occurrences on NOMAD non-cancer V3. Once we've got a set of rare variants, we then look at splice prediction tools. And we're currently using Splice AI and Pangolin, both publicly available from the Broad. And we set a threshold for sequencing with a prediction of 0.1. And I'll go into that in a little bit more detail. So far, from the 120 families, we've identified 95 rare deep intronic variants across the 10 genes. And when we did our Splice AI and Pangolin analysis, only 7 of those variants had scores greater than 0.1, and they always predicted a gain of a donor or acceptor, not a loss. The 7 deep intronic splice variants were evaluated by long-read cDNA analysis using the Nanopore's direct cDNA kit. We used patient whole blood RNA, total RNA, and in some cases we had lymphoblastoid cell lines to use. And we reverse transcribed with gene-specific 3' UTR primers spiked in, and they were designed to the 3' UTRs of each of the 10 genes. So we had a pool of 10 genes to try and enrich for transcripts from those 10 genes. So this is the first variant that we identified, and it was actually in 2 different families, 3679 and 6196. It is located 1.3 kb away from the end of exon 16 in BRCA1. Interestingly, both of these families are from Sicily. So this is a potential founder allele. So if there are any Sicilian geneticists in the audience, please reach out to us. We might be able to track down more families. As you can see here, the variant is represented by a red V, and it was first seen in the 2 sisters from family 3679 diagnosed with breast cancer in their 30s. And then the proband of family 6196 was diagnosed with breast cancer at 42, and the variant was also present in her cousin who had bilateral breast cancer at ages 29 and 36. So the first thing we did was genotype the additional family members that were available to us in the study, and you can see the variant co-segregates with breast, ovarian, pancreatic, and breast cancer in both extended families. So I'm now going to show you a bunch of figures that are set up like this, with the gene structure on the top, our long-read genomic DNA reads underneath, showing the variant detection, and then our cDNA long read showing the gene expression for this particular region. So as I said, this is a single base change, 1.3 kb from the end of exon 16. It was predicted to introduce a new donor site. And when we did the cDNA analysis on the proband of family 3679, you can see there is a pseudo-exon that's expressed in about half her messages from her blood RNA. We sequenced that, of course, and it's a 74 base pair pseudo-exon. And when it's spliced between exons 16 and 17, it introduces a premature stop codon. So it's therefore a pathogenic variant and explains the extremely high cancer risk in this family. Our second BRCA1 deep intronic variant was in a family of Mexican ancestry. The proband was diagnosed with triple-negative breast cancer, and her sister had ovarian cancer. This was 473 bases away from the end of exon 14, and it again was predicted to create a new donor site and introduces an 84 base pair pseudo-exon, which again, when incorporated into the BRCA1 message, introduces a stop codon. Our third example of the BRCA1 deep intronic variant is it's not actually that deep. It's just actually 69 base pairs away from exon 16. And again, the proband here was diagnosed with triple-negative breast cancer, and as you can see, her mom, aunt, and grandmother had ovarian cancer. And when we zoom in on the cDNA, the long-read cDNA reads, you can see that rather than generating its own pseudo-exon, this splice change allows the exon to read into the intron for 65 bases. So that in effect, the exon is extended. But there are multiple stop codons within that intron. So again, this introduces a premature truncation in BRCA1. The next three examples are going to be in the ATM gene. This first one was identified in a patient diagnosed with metastatic prostate cancer. We haven't been able to genotype the other family members yet, but we're trying to recruit them. And this actually was the lowest score that we saw on Splice AI. It was a 0.015, so just below our cutoff. And this was actually interesting because it creates three different transcripts. The first line represents the pseudo-exon that was predicted by Splice AI. But then if you look down to the second cDNA line, you can see there's actually another pseudo-exon in the intron before exon 57, which we didn't anticipate because it's kind of a long way from the normal splicing machinery. And then the third transcript that we saw is that pseudo-exon from intron 56, followed by a complete intron inclusion event. So the full intron from exon 57 to 58, which is about 3 kb, is incorporated in that message. Another example of an ATM variant is in this family, where the proband was diagnosed with clear cell ovarian carcinoma. And again, this is a new donor site with a pseudo-exon. And then the final family I'll show you actually came to us because they were diagnosed with ataxia telangiectasia, indicated by the gray box. This is a recessive neurological disease caused by heterozygous pathogenic variants in ATM. And the clinicians who approached us, they knew about one of the pathogenic variants. It was a canonical splice variant that caused exon skipping, but they hadn't identified the second variant. So we did adaptive long-read sequencing and found a deep intronic splice variant that was only 36 bases away from the variant in family 5431. And it actually activates that same cryptic donor site and introduces a premature truncation. So we looked more closely at the ATM literature, and there's actually a third variant on ClinVar between these two variants. So that's 3 pathogenic deep intronic variants within about 30 bases. So we thought this might represent a hotspot or a clustering. So we looked more closely at that specific region. And I'm showing here the 2 variants that we identified. The third variant is between these. And what's interesting about this is on the antisense gene in this intron is a gene that's expressed in the opposite orientation. It's a CORF62. And we're kind of speculating that the branch point of this antisense gene may contain cryptic sites for cryptic splicing, and we're going to look at other genomic regions where there's an antisense gene. So in summary, our approach combines adaptive sampling to readily identify deep intronic rare variants, splice prediction tools to help prioritize which events to follow up, and then long-read cDNA sequencing to look on and validate the effects of the splice variants. So of the 120 previously unsolved families, we think we've explained 8 or 6% of them with rare deep intronic variants in BRCA1, ATM, and we recently identified a PALB2 variant as well. So moving forward, we are hoping to multiplex samples more deeply, and we're very excited to hear about the improvements in adaptive sampling last night. So hopefully we can potentially run 6 or 8 samples for a flow cell now to reduce cost and increase throughput. We'd also really like to try and refine the splice prediction tools, particularly for small insertion deletions. We haven't really tackled that yet. We've only tackled SNVs. And then finally, we're really excited to incorporate direct RNA sequencing into this analysis. And I would like to thank Suleiman, who did all our bioinformatics, Amal Abubarayan, who did all the RT-PCR and Sanger confirmation of the pseudo-exon events, Jessica Mandel, who is our genetic counselor and does all the outreach to these families, coordinating the genetic testing results and getting us blood samples when needed. And then, of course, Mary Claire King, who's been working on inherited breast and ovarian cancer genetics and genomics for more than 40 years now. Thanks so much.